Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kaim Biology. Today in this video lecture we are going to talk about initiation of DNA transcription in a detail. So let me write. Initiation of DNA transcription. Now, in the previous video lecture, if you guys remember, we already talked about the summary, the overview, the big picture, or the concept about the DNA transcription. And we also talked about the enzymes and the protein which are used. So we talked that there is only one enzyme which are used in DNA transcription in prokaryotes, and we called it Rna Polymerase 2. And we have only one protein also, which we call it Rho protein, which are mainly used in the termination of DNA transcription in prokaryotes. Okay, now if you miss that video lecture, it is present into my channel. You can go back and watch it, and then you will understand about the initiation. Before that, you need to understand the overview and the concept about the DNA transcription. So I already explained it. You can watch it into my channel. Now, what is initiation of DNA transcription? Initiation is that stage in DNA transcription where the DNA transcription is started. Okay, where the RNA synthesis is ready to start. This is called initiation of DNA transcription. Okay, now how the initiation of transcription will be carried on or inside the prokaryotic cell. So for that we need to understand the promoter region, because in a DNA molecule, we have a specific region which we call that promoter region. At promoter region, the RNA polymerase, which is specially responsible for the DNA transcription, so where the promoter are present and where the RNA polymerase can bind on the promoter and how it can start the initiation process. So we need to understand about the promoter, the main sequences which are found in promoter, and we also need to know about the upstream and downstream sequences or element which are found in the DNA. Now once we get idea about that, then the initiation of DNA transcription will be very easy. So first of all, let's try to understand each of the terms as I told you. Now let's suppose, now to understand each of the term, let me draw a DNA structure here. So we already know the upper strand is the coding while the lower strand is the template strand. Okay. Now in the DNA strand, we have a promoter region somewhere at that specific point. So let's suppose this is the DNA molecule. So at that specific region, at that position, so there is the promoter region. And after the promoter region, there are some other sequences which we call that +1 position. So this is our +1 position, and we call that RNA starting point. From there the RNA are synthesized. Okay, now that area is called as promoter region, and at that specific region the RNA polymerase 2 can bind here. And remember, RNA polymerase 2 is a very huge protein, is a very large enzyme as compared to DNA polymerase. It can cover the whole DNA, means it can cover the promoter as well as the starting point as well. It is too much long. It not only cover the promoter, somewhere the promoter region it can cover. So just think about that, how much this RNA polymerase will be in the how much it will be large. Now in the promoter region, there are some consensus sequences are present. Consensus sequences means that there are some sequences which are always present in every DNA, in every organisms, but a little bit changes maybe occur. Now what kind of sequences are present in the promoter region? In promoter region we have -10 position and we have -35 position. Let me draw here. So at that specific position there will be -10 position, and at that specific region there will be -10 position. and after that there will be +1. So what are the concept behind that -10, minus and plus? What are the concept behind that? Remember, at that position which we call it -1, we also call the +1, we also call that 0 position. So remember, when the sequences or the elements or the nucleotide which are present somewhere at that position, after the +1 position, the sequences or the element which are present after the +1 position at the right side, according to me at the right side, then we call that downstream element or downstream sequences. Stream, and it will be written as +1, +2, +3 and so on. And remember, when the sequences which is present before +1, okay, before +1 means at the left side according to me, then we call that upstream, means at that position. So we call that upstream element or upstream sequences. Okay, so remember, the promoter is always present at upstream area of the DNA. As you can see, this one area is called promoter, so it will be always present at upstream. Now this -10 have a consensus sequences, and these sequences are usually we have TA, TAAT. We also call that TAT box, and we also call that Prinbo box. Okay, so at -10, at -35 position, there are some other consensus sequences. Let me write A, C, A. Now remember, these sequences are present at this lower strand, 3 to 5 prime. So their complementary will be, you can write their complementary. So these are the consensus sequences that are usually present in the promoter region. Okay, this -10 is actually the transcription start point, or we can say initiation of transcription start point. Okay, this -35 will be transcriptional rate. So at that specific position, the transcription will be going fast or it will be going slow. So it depends upon on 35 position. On -30, on -10 position, the transcription initiation of transcription are started. So this is all about the promoter region and some of the sequences which are mainly found in the promoter region. Okay, one thing more. Promoter, there are many different promoters which are present in E. coli cell. As we are talking the DNA transcription in E. coli cell, so remember, we have different promoter at different position in E. coli cell. So it will be our example. So I write the promoter here as an example. One thing more about that, DNA transcription in case of the prokaryotes is a de novo. What does it mean? It means that as we talked that in our previous video lecture, if you guys remember, a DNA replication, if you miss, it is present into my channel. In DNA replication, what we talked that for DNA polymerase enzyme, they need some of the RNA sequences which we call that ribonucleotide sequences or primers, due to which they can start the elongation, due to which they can start the process. But RNA polymerase don't need any primer. Okay, they don't need any primer, so they will start the RNA synthesis without needing any primer, which we call a de novo synthesis process. Okay. Now remember, at promoter region, the RNA polymerase should be bind tightly. Okay, so we have RNA polymerase 2. So here I'm again not drawing the exact structure of the RNA polymerase, but somehow it will be look like in this form. So let's suppose this is the RNA polymerase structure. Okay. So this is the RNA polymerase 2 structure having different subunit alpha, beta, gamma, alpha, beta and omega, means have mainly 5. And this RNA polymerase, let me write RNA polymerase 2. And this RNA polymerase 2 are mainly in a deactivated form, and it cannot recognize the promoter region by itself. Okay, it is very difficult for RNA polymerase to recognize the promoter region, and it is very difficult for the RNA polymerase to bind on the promoter region so tightly to start or to initiate the transcription. So for that purpose, it need a factor, and we call that sigma factor. So most of the time it need a sigma factor at that position. Let's suppose this is the sigma factor. So when the RNA polymerase to bind with the sigma factor, so this RNA polymerase is now become activated. And remember, without RNA polymerase, without sigma factor, this RNA polymerase is called as coenzyme, which is in a deactive form. But when they got sigma factor, and when the sigma factor are bind with it, then this coenzyme is converted into whole enzyme, which is the activated form of the RNA polymerase 2. So let me write here, it will be converted into holo enzyme. And now after that, it will tightly bound on the promoter region, and then it will start the initiation of DNA. It will start the initiation process. Okay, so how it can bind here? So again, let me draw the DNA here. So let's suppose this is again a DNA structure, and this is, let's suppose, the promoter side. So here I'm not writing everything about the minus 10 and minus 30. So let's suppose I'm not writing here again minus 10 and minus 30, but let's suppose this is the promoter region. Promoter. Okay. And somewhere at that position, there will be +1, means RNA starting point. Now, sometime what actually happen? Most of the time the RNA polymerase 2 will bind here, as you can see. So let's suppose this is the RNA polymerase 2, they are bind at the promoter region, not at the promoter region, before somewhere at that specific position. At that specific region, the RNA polymerase 2 can bind here. Now after that, it will need a factor. So when the sigma factor, let's suppose the sigma factor are binded with this RNA polymerase 2, when the sigma factor bind with it, so then it will bind. Now it is bind very loose on the promoter region, but when it bind with the sigma factor, so then it become activated and it bind with the promoter region tightly. One thing. Second thing is that due to this sigma factor, the RNA structure is a little bit modified, means the RNA structure is a little bit large, so elongated. The RNA polymerase structure are now elongated, become elongated. So in the next diagram, if I draw here again, so how exactly it will be look like? So this is the RNA polymerase structure, and there will be the +1 position, as you can see. And this is the promoter site. Let's suppose here I write just P. Now, due to this sigma factor, the RNA polymerase are become enlarged. Now, why it become enlarged? What are the reason behind that? It become enlarged because they have to bind tightly on the promoter region, and they have to recognize the promoter region so tightly. That's why they enlarge their self. And due to this way, they almost cover 60 to 80 base pair or 50 to 70. I don't know the exact, but suppose they most of the time cover 60 to 80 base pair. Okay, and remember, when sigma factor and RNA polymerase structure is changed, then it make a complex which we call that closed binary complex. We call that closed binary complex. Let me write closed binary complex. Okay, so this is called as closed binary complex. Now one thing more about that. Remember, with this sigma factor, the RNA polymerase cannot be moved and they cannot start the initiation process with having the sigma factor. So what actually we get here, we get the point here that this sigma factor should be released. Once the sigma factor are released, so then the RNA polymerase will start the transcription process, or they will start the initiation process. Okay, so this sigma factor will be released out. So after some time, what actually we get, this sigma factor should be released out. So here again, let me draw the RNA structure here. So this is again the DNA molecule, and let's suppose this is the promoter site, and this will be the +1 position. So remember, when the sigma factor is released out or removed, then the structure of RNA polymerase are again small, or it again back to its original position. So again the RNA polymerase are become small, or it can reach to its original position again, as you can see, because the sigma factor are released from here. So now the sigma factor are released out. After that, at that +1 position, the DNA are means some catalytic process are starting here. Due to this catalytic process, the DNA are melted. The DNA strand are separated from each other. So after that, we get the structure like that. So again, okay, this is the promoter region, and this is the +1. So at +1 position, the DNA are become melted, and these are the sequences, DNA sequences, as you can see. And this is the RNA polymerase 2. Okay, so remember, when the DNA are melted at that specific position, and when the sigma factor are released out, then it make another complex. We call that complex open binary complex. So binary complex. Now after some time at open binary complex, what happen? After that, the RNA synthesis will be started. Some of the RNA sequences will be added, ribonucleotide sequences will be added at that specific position. And when the RNA synthesis are started, few nucleotides, 4 to 6 nucleotides are added here. Then it make another complex which we call a ternary complex. So let me draw here. So let's suppose again this is the DNA strand. So this is the RNA polymerase 2. And these are the DNA sequences, as you can see. So this is the promoter region, and that one area is the +1 position. Now at +1 position, as we already talked, so at that specific area the RNA synthesis are started. So few of the RNA nucleotide are added here, RNA. Okay, now at that specific position, when the RNA synthesis are start, so we get another complex, and we call that ternary complex. So let me write ternary complex. Okay, what happened after that? As we talked that when they have the sigma factor, it will be tightly bound. So let's suppose the sigma factor, as we know that sigma factor is all means released out. No, the RNA is moving. Remember, at that position the RNA is not tightly bounded. So when the RNA is not tightly bounded, then what happened at that specific position at ternary complex? The RNA is start and synthesizing this RNA polymerase is ready to start the synthesizing the RNA strand. So when it start 4 to 6 nucleotide addition, then this RNA polymerase will be fall down, and again it will come back to its position and again make the RNA synthesis and again start the RNA synthesis, and it will again fall down and then come back to this position and again start the position, and this process will be repeated so many times. 4 to 6 times this process is repeated, 4 to 6 times. So at that process the RNA are fall and come back to the promoter region and start the synthesis. So what actually we get? We get many of the RNA strand pieces. So as you can see, so this is many of the RNA strand pieces. So many of the RNA pieces we get. Now this process is called abortive initiation of DNA transcription. This is called abortive of DNA, initiation of DNA transcription. Because why the RNA are falling here? Because the RNA cannot bind here tightly. Why? Because the sigma factor are not binded here. Then after 4 to 6 times after that, the RNA are bind here tightly, because now they get their position tightly here, and after that the RNA polymerase are not falling here, and then RNA polymerase are moving and start the elongation process. But after 4 to 6 times falling, after that they will start the elongation process. Okay, so how the elongation process will be started after that, so we will talk it inshallah in our next video lecture. So this is all about the initiation of DNA transcription, that how it will be started and what are the main sequences and elements are found here. So I hope you get idea about the initiation of DNA transcription. Inshallah in the next video lecture we are going to talk about elongation of DNA transcription in a detail. So please stay tuned and keep watching my videos. Thank you.